So I'm an assistant professor at University of Pennsylvania, but before that I was actually here at NIH for several years at NIAID. And so I've done trials here at NIH, and then I'm involved in clinical trials both on the investigative team and also on data safety monitoring boards. So we're talking a little bit about from my own personal experience and also just from the general wisdom about data safety monitoring. And I do want to acknowledge many people have contributed to this presentation over the years, including Laura Lee Johnson, who I think has taught a few other lectures here, Dennis Dixon, Michael Proshan, all either formerly or currently at NIH, and Susan Ellenberg at UPenn, who used to be at the FDA. Long list of experience, adding to a few of these slides today. So I'm going to give an introduction to this topic, and I'm going to just pretty much almost immediately launch into a couple of case studies or a couple of quick examples, because I think you really understand the what, the why, and the how by seeing examples for this particular topic. And then we'll cover the basics of data safety monitoring boards, how you form them, what they monitor, and the basic principles, when you need them and when you may not need them. And I'll finish with just some specific examples of things that are monitored by the DSMB, so going from the broad to the concrete through this. So the fundamental principle guiding data safety monitoring boards and overall concept of data safety monitoring is you can imagine you're doing a clinical trial of a treatment. It's novel, you don't know if it works, it might have some risks. You don't want to wait until the end of the trial to figure out whether or not during the trial something fundamentally went wrong with the conduct of the trial, essentially making the results unusable, or that the new drug had unexpected harms, or that there was convincing evidence very early on that you could have stopped and given this treatment to people much earlier than you did. Trials can go on for years. So ultimately, the idea is that clinical trials, it's an endeavor that's very important, but it's not without risks, and we want as many safety measures in place. And so data safety monitoring boards exist for that caution. Another fundamental concept is once you start, say, looking at data at interim looks during your trial, you can't just do that without any restrictions if you want a valid study at the end. And hopefully you've gone through some of these lectures on hypothesis testing, on randomization and maintaining the blind. Everything we know about clinical research really comes to a head in the concept of data safety monitoring boards. So one of the things that we learned from hypothesis testing, which I believe was already on your schedule, is that if you look at the data multiple times, you increase the risk of a false positive, right? So you flip a coin with a 5% chance, eventually it's going to come up heads. And so if you think you need to look at data in interim looks because you want to make sure that if you need to stop the trial, you stop it, you need to have a statistical plan in place so that you do that and at the end still maintain the statistical validity in your analysis. That you want to maintain that acceptable rate, say 0.05 or whatever your desired rate is. It could be 0.01 for certain trials. That you maintain that no matter how many times you need to look at the data in the interim. And that's possible, but you need a data safety monitoring plan. Essentially, once you have interim looks planned, even by an independent body, you need a statistical plan that needs to be part of your protocol, generally preferable from the outset, from the design. And it also, you realize if the goal is to be looking at data in interim looks, then your data safety monitoring board is going to need people that can consume that data, that they're competent in understanding the difference between unlucky streaks in the data or statistically convincing trends, and that they can understand all the complexities of clinical research, including risk benefits and anything else that you might need to consider in order to make the decision of whether the patients are best served by continuing the trial or stopping the trial. And finally, if you have a group of people now charged perhaps at looking at data, or the only ones that may be unblinded, you need them to be objective, right? If they're going to be having this responsibility of deciding is it safest for the patients, is it best for to continue or to stop, they can't have any vested interest in the trial continuing, right? Or perhaps some relationship with the study investigators or investment in the company or So suddenly you realize, based on the responsibilities of this board, that initially we started with, well, we just want to protect patients, quite quickly becomes a very complex endeavor where you need to consider very carefully the people on the board and their responsibilities in order for them to be able to fulfill their responsibilities. So this definition comes, I think, after many years of trials, from their infancy, just people doing them, to having quite structured guidance from various bodies on when and how you create a data safety monitoring board. So the basic definition is that it's a group of independent experts that reviews the ongoing conduct and involving data, so emerging data of a clinical trial, to ensure continuing patient safety, as well as the validity and scientific merit of the trial. So not just that the data is not showing any early signals, but the trial that is being conducted is the trial that was planned, that the protocol is being faithfully followed. That's another sort of part that perhaps I haven't initially introduced, but I'm introducing now, that essentially that this trial is as intended and is ethical to continue. And this is just a brief note on terminology, because if you start reading, right away you start seeing that there are DSMCs and DSMBs, and mostly that's a cultural thing. Europe and the FDA tend to use committees, DSMCs, and that NIH uses the Bs, so Data Safety Monitoring Boards. I can just remember early on being confused if these were different types of bodies or are they the same body, but it's just general preference. But what's interesting is these groups are entrenched, and sometimes when they collaborate, they argue over what term to use. In any case, let's get into our case studies. So the first example, I think both of these studies that I'm going to talk about are historical, but incredibly exemplary in the sense of being the poster children for why you need a DSMB and the benefits of having a DSMB. So the first one is ACTG 076. So that's the AIDS Clinical Trials Group Protocol 076. And this is from the early days of HIV. And cidovudine was just recently shown at the time to be able to slow progression of HIV. It was the first therapeutic breakthrough and the first treatment approved by the US government to treat HIV. So the trial on the docket that was going to be done was to see whether or not there was safety and efficacy in using cidovudine, or AZT, in preventing transmission of HIV from infected mothers to their infants. And in particular, these women couldn't have advanced disease. And this trial, I give the results paper, which is the reference there. So at the time, NIAID had a group that basically ran all the trials that NIH was launching funding for HIV, and they had a DSMB that was monitoring several of these trials. And so there were various ethical considerations from the get-go that they had to think about in designing this trial. And the first is, if partway through, they already have evidence that this works, that AZT works in general at slowing the advancement of HIV, there's a good chance that that could reduce infectivity of the mother. And so if you want to stop as early as possible, once you have convincing evidence, right, that you can prevent babies from being infected. This is a very grave diagnosis in the early 90s. So that's the first ethical principle that needed to be in the design of this trial. And the second was there was a lot of pressure to just give drugs immediately, but you didn't want to put out a drug that didn't actually work. And particularly for pregnant mothers and unborn infants or newborn infants, there are some extra risks there that we don't quite fully understand when we have new drugs given to pregnant mothers. So we wanted convincing evidence that there was overall benefit and not undue harm. So, that becomes a statistical analysis, and it becomes interim analyses that a body can look at during the trial. Basically, instead of waiting until the end, they're going to look at different times during the trial at these issues of what are the adverse events and what are the relative event rates of infection on those on a placebo versus an active arm. So it was powered to have an 80% chance to detect a 33% reduction of HIV transmission through the first 78 weeks of a baby's life. And so that was relative to a projected rate of 30% in what they knew about transmission at that time. So the target sample size was 748, and accrual began in 1991. So you might remember from the survival lecture at the end of last year, when you have time to events, time to transmission of HIV, babies getting infected, the sample size is the number of, typically the number of observed events that you need in order to determine the power. And so you have the idea of how many do you need to accrue to get the total number of events. So accrual began in 1991, and the projected accrual was to take at least 5 years. And actually, now that I'm reading this slide, remembering the 748 is the number of mother-baby pairs you needed for the expected event rate to have 80% power. So already you have to do a little bit of a guessing game for sample size, which is another reason why you might have a DSMB look partway through to make sure you're on track in terms of your projections and having adequate power. So the projected accrual was to take 5 years, and they were planning for 15% dropout. So the monitoring plan in place was to meet twice a year just to look at safety. Automatically, that was happening. And in addition, they were going to do efficacy reviews, and that was after each third of the projected infant infections. And then they projected that the first efficacy review should take place in order to capture about a third of the events around February of 1994. And so then that would happen, this is a little bit of the nitty-gritty. You can't just say, okay, we'll use all the data up through January 31st, right? So they basically locked the data for all mothers enrolled up to December of 1993, and then they would look at the events up to that point. So this is the data analysis at the first look. And so what we're looking at here is sort of the opposite of a Kaplan-Meier curve. So normally survival curves, so you look at the event rates. Nobody has the event, say, mortality at the beginning of the trial, and then they decrease, and then you can compare the mortality rates over time or the percent alive. Here what we have is, some babies might be born basically instantly infected, and so you'll see that the event rate, let's get this going here, is not quite at 0. Maybe it's a little bit higher. So babies are essentially born already. There seemed to be an apparent difference. And as time went on, the zidovudine arm only had 8% of the babies infected, whereas placebo was projecting 25% by the time a baby got to 72 weeks. That's a more than fourfold difference. And the p-value was really convincing. And at this point, if you look at the total number enrolled, it was about 360. And I think basically they had about half of the planned events that they were expecting. And so quite a lot of information had accrued. Events and accrual had happened a little faster than expected. And so this was overwhelming evidence and certainly met some conditions where you might think, well, okay, we definitely need to stop. We have evidence of benefit. But there were some discussions. And so the first discussion was that they knew there were 46 babies that were sort of in limbo. They didn't know their HIV status, and they didn't want to stop for efficacy and then have those babies evaluated and realize retrospectively that the boundary wasn't actually met for stopping. And so, because there is more evidence that's needed, not the usual 1.96, and we'll get into that in a little bit, when you're stopping early. And so there was some statistical analysis to look at reasonable trends of what could happen to those 46 and what could happen to the p-value depending on different outcomes. That was considered. They looked very carefully at safety. And that type of analysis that I'm talking about, just to explain all the words on the slide, is what we call conditional power. So conditional power is essentially, given the data up until this point, what's the probability that you will wind up with a positive result at the end? And so that is often a statistical analysis that will be presented to the DSMB when pertinent, when you're starting to think about, is it time to stop? So after careful review of that conditional power, of the safety, of how much data was accrued, which was quite a lot, the DSMB did recommend that there was overwhelming and convincing statistical evidence that there was benefit to this treatment, and they recommended the trial stop. And a key term here is the DSMB is a board that's an independent body. They're not the sponsor, they're not the investigators. They give advice, guidance. But in the end, it's the trial leaders and ultimately the sponsor that will make the decision, typically on whether the trial should go or no go. And in this case, as in many cases, they followed the advice of the DSMB, the recommendation, and they stopped. And instantly, this zidovudine was provided to those in the control group, and guidance in the US and across the world was changed to allow this to be a treatment for prevention. So that was a great success, and the benefit of having the DSMB was that they didn't have to wait to the trial's end. They got to stop actually a couple of years earlier and get that treatment out. So many babies were able to escape infection because of that earlier stopping and distribution of the treatment. So here's a different kind of example, and exemplary in another way, and it's CAST. And there's actually many, many lessons that can be learned from this trial, as is typical when you do a large trial. So this was the cardiac arrhythmic suppression trial, and it was designed to evaluate a hypothesis of whether suppressing ventricular arrhythmias in patients with recent MI or heart attacks, whether or not that would reduce sudden death, that was the primary endpoint, or overall mortality as a secondary endpoint. And there were actually several active drugs that were known to be successful in suppressing arrhythmias, and so this trial looked at 3 of them, econide, flecainide, and morticiazine, which a little more shaky about how to pronounce that one. You can see it on the slide. So we had 3 actives and 3 matching placebos, essentially a large, complex trial. And they were randomized 4,000 patients, partially because they were studying several treatments at once, also partially because the need for that event rate. Those are the number of patients you randomize, and then not all of them will wind up with those events. And as we learned, it's the number of events that's your true sample size. So they needed to randomize 4,000 for 90% power in the end to detect a reduction in sudden death with a 0.05 one-tail significance level. So that was the original design. And I don't know if any alarm bells are going off. How well Laura Lee jumped up and down about this. It was Laura Lee Johnson who did the hypothesis testing. Yes. But so the DSMB met for the first time. So this cardiovascular trial was launched in 1987. The first meeting of the DSMB, so this was a blinded trial, large trial, death as an endpoint. It's going to have an ethical monitoring board as an independent. Phase 3, you're basically required to have a DSMB. Their first review was before even the study started, and that's often the case because they want to look at the protocol. They want to make sure that this monitoring board is okay with the protocol as is and agree on their charter. What are they going to monitor? Is everyone on the same page about what's appropriate to monitor and how? And as a result of that meeting, they made a recommendation, actually, to change the primary analysis of the trial. And actually, 1987 now is going on 30 years ago. And statistics was getting, in clinical trials, was still sort of being formalized in terms of what are best practices. And perhaps this is a time when it basically became convincing, and certainly there was a forward-thinking statistician on that DSMB that just was no longer acceptable to use one-sided 0.05 level tests. So the initial argument is we know the smaller the alpha, the more difficult it is, the more N you need to guarantee that low of a false positive rate. And someone might argue, why would I want to spend alpha to look in either a strong positive direction or a strong negative direction to decide whether or not there's a treatment difference when I'm only interested in the strong positive direction of benefit? I want all my alpha going to the 0.05, and then I can use 1.64 and not 1.96. But the opposing argument that has become the dominant one is that you don't want a test, whether it's one-tailed or two-tailed, to influence the level of evidence that you're going to use to declare benefit. Right? So whether you can do a one-tailed test, but it shouldn't be easier to pass that test than a two-tailed test in terms of deciding what is the benchmark, what is the critical value, in this case 1.96, to declare significance in terms of a difference between two groups. Say if you're doing a one look at a 0.05 level. And another argument is that, you almost rarely, I've never seen a one-sided confidence interval. When you do a confidence interval, you do the uncertainty, plus or minus, say 1.96 standard deviations around the mean. If you do your test as a one-sided test and a confidence interval as a two-sided confidence interval, you could wind up doing this test, declaring a difference, having a confidence interval, and having the null inside of that confidence interval. So it's, some might view, if you're more on the clinical side, this is like an esoteric argument. Why aren't the statisticians on board with this? Well, they are now, but at the time, perhaps it was an emerging consensus. And it was interesting that at the very first meeting, essentially the principal analysis was changed. Well, they made the recommendation, and then the study team and the sponsor were convinced by the arguments, and they did change it. And it only reduced the power a little bit. So they were already going for 90% power, which is quite convincing. So they were still at 85% power when they did this change to be two-sided. So I think in the end, everyone was satisfied with that change in the protocol. And so the lesson learned here is it is very important to have that first meeting before the study starts. Because ultimately the DSMB is charged with a pretty delicate sense of responsibility, actually a very important role, which is they are going to be looking at data that no one else has and be making recommendations that have a strong influence on the future of this trial. Everyone needs to be on the same page scientifically, believe in the trial, believe in its validity from the start, and then be able to trust each other and get along through what could be potentially difficult times in terms of unexpected challenges that make this trial more difficult to complete than anticipated. So you really want that first meeting so everyone is on the same page about the roles and responsibilities and the science. Another lesson learned was not only should you be on the same page about the design, but the monitoring plan should be in place. Because what happened at CAST, which I think was just one of those every 6 months, just basically there are two kinds of meetings the DSMB has. One is just regularly scheduled, we'll just keep an eye on safety, and then you'll do these interim looks at efficacy as you go, not expecting real efficacy trends until a lot of data has accumulated. But in this trial, with only 5% of the expected information, 5% of the total number of events that they needed for that power, they actually already had a difference apparent between the treatment arms. Now, as is typical in a DSMB report, once you start looking at efficacy, the report is going to just have arm differences, and you might have them labeled as arm 1 and arm 2. Because you never know, you want to be, out of preponderance of caution, you want that report to be masked in case it gets into the wrong hands, mailed to the wrong person accidentally, left on a desk. But then the DSMB would have the potential to say, okay, tell me which arm is which, because I want to make a decision. And others will always have that which is which, say in a separate envelope that they open up at the start of every meeting. In this particular trial, as was done at this time, more commonly than it is now, they chose to remain blinded until perhaps they decided there was a reason to look. And so they looked, all they could see was there was a treatment difference. At the time, they hadn't had stopping boundaries in place, perhaps because it was so early and they were still gelling the analysis plans. And so in the end, it was probably fine, because even though there was a convincing, when they did the test, in this case it's a log rank test, because they're looking at survival curves between inactive and inactive, or say placebo, the log rank looked like it was exceeding. It was 3.43 for a treatment difference, and the boundary that they were eventually applying, retrospectively looking back, it seemed that they had exceeded it. But it's that issue of there were pending events, and actually when they were adjudicated and added in, it actually would have flipped it so that it would have been less convincing. So in the end, even if they had the boundary in place, even if they had looked to see which arm is which, they may not have stopped. But it was instantly alarming. But they chose to remain blinded. And at the next look, and there's a reference there for this trial that people talk endlessly about for many reasons. At the next look, there were 48 events. So at the first look, it was roughly 22. Now there were 48 events. And actually looking at event rates and other reasons, they realized that they would only expect 300 and not the 400 and some they'd originally thought at the end of this trial, which meant that 48 was a little bit higher of a percent in terms of total number of events. And when they looked at the boundary, they saw that their log rank was actually still at 3.22, and it was well over the boundary for stopping, plus or minus 2.98. And so the DSMB requested to be unblinded to find out which arm was which. And much to their surprise, it was the placebo that was doing the best. And it was active arms that were having harm. And all along, everyone had assumed it was benefit, that just they were waiting for it to be convincing. And this becomes the compelling argument for why most people argue you should not say, well, to remain objective, I don't want to know which arm is which until I really think there's a difference. And the reason is you're probably not going to make a symmetric, even decision in terms of how much evidence do you need to convince the medical community to adopt this new therapy, which might have harms as well as benefits, versus evidence to say, you know what, I'm 80% confident that we might be harming people, but I think we should wait till 90% confident because it's kind of early. No, so it's really not a symmetric operation. And I think having seen this over and over time, people have just, a lot of the think tanks that make recommendations on how to do these things, people who have served on these bodies many times in their careers, make an overwhelming recommendation that the DSMB is not masked. Trial is double-blinded, the investigators and the patients. It is not triple-blinded. We do not blind the DSMB. We want the DSMB to have every bit of information possible to make the decisions. And this is ultimately why. Because you can't help but sort of believe in the new treatment. And in fact, they were so convinced that something had to be wrong that they investigated, they got random bottles. They assumed maybe the place where they were labeling the bottles had made a mistake, and that the placebo was the active and the active was the placebo. So they started testing chemically bottled medications that had those labels. They looked for imbalances in key variables in the placebo and the active arms to say maybe the differences were because these folks weren't actually have the same risk factors. And then they continued to also look to see if there were certain subgroups that maybe were being harmed versus others, again thinking, well, if that subgroup was out of balance, that might have driven this treatment difference. And they also looked at the secondary endpoint, which was overall mortality, to see if it was consistent. And finally, when they realized it was in fact the placebo that was doing better, they just wanted to see whether certain arrhythmia drugs out of the 3 that they were studying that were actually the problem. And it turned out early on that there were 2 out of the 3 active drugs, econide and flecainide, luckily the 2 I can pronounce, that were driving the difference. So each of the 3 actives had a matched control, and so the overall score was 35 events on the active, 13 on the placebo, and when they reduced it down to those on the econide and flecainide versus placebo, it was 33 versus 9. So what had just been CAST became divided up into CAST 1, which was the trial with all 3 treatments, and then at the end of this DSMB, they stopped the econide and flecainide arms, but they continued the third active and matched placebo because there was evidence, certainly there was no reason to stop those arms. And so that was the benefit of having sort of the structure of multiple matched placebos, that they can continue the one question even though they had to stop the other 2. So other lessons learned here is that not only should your monitoring plan be in place, your DSMB be in place all before your study starts, but your data management system, your data entry, all those people in charge of monitoring your data, entering your data, organizing your data should be on board on day 1, and that data system has to have been built and tested because you need that data at that first meeting if it's available, because very early on trends can become apparent, certainly for harm. And also, it turns out, for benefit, say in the ACTG trial. And other principles are, you need to be organized, you need to have these meetings on the books, because there could be many reasons why you need to act quickly, so start your monitoring right away. And also, you need a board that's going to be able to think about, and investigators, contingency plans about what to do once you have this information. So people need to be ready to receive the DSMB recommendations as well. And I think, as I mentioned earlier, I think probably went over most of this, is that the ability of the DSMB to do their job can be compromised if they remain masked. And I think initially that idea of objectivity seemed to be the overwhelming principle of why we would keep the DSMB masked, unless, and perhaps always give them the prerogative to ask to be unblinded. But I think it was, examples like this, that in general, I think the recommendation is that you keep a DSMB body unblinded. The report may stay masked because you never know who could intercept it, but you would always have that DSMB have the ability to see which arm is which. And a great article, and it's just an editorial, it's only about 2 pages, that really summarizes both sides, I think, but it's definitely by the name of this article, Mass Monitoring Clinical Trials, Blind Stupidity, ? Obviously coming down on one side of the debate here, and certainly a good way to augment reading if you're interested in this topic. Because they do present the opposing view, at least briefly. Okay, so hopefully those 2 examples, sort of have given you some ideas of why we need a data safety monitoring board. Essentially, to summarize those examples, we need to be able to identify any safety problem rapidly. We need to be able to identify logistical problems, if there are some, so that the board can recommend amendments or make sure that those come to light, such as, if the accrual is so slow, they'll never get full enrollment by the end of the grant period. That could be a problem, so maybe you need to add sites. So they'll be actually evaluating the feasibility of the trial, not just things like efficacy or safety. And ultimately, they want to make sure if the trial objectives have been met early, then the trial should be terminated early.